Thank you for the introduction, Homan, and also for the opportunity to come here and present AlzeCure Pharma for you here today. Now, AlzeCure Pharma is a Stockholm-based company and founded by a group of drug discovery professionals with extensive background from pharma industry. Our key focus is on Alzheimer's disease, which the name implies, an area of huge unmet medical need. And we target this disease using two different projects. One is called NeuroRestore, which targets the symptoms of the disease. And the other one is called Alzstatin, and that's targeting the progression of the disease, both of them being small molecule therapies. Now, as you know, Alzheimer's disease is a disease that affects the brain, which is obviously the most central organ in our body and who makes us who we are. Now, the brain has several different areas that are communicating with each other in enormous and vast networks of nerve cells that send electrical impulses between them to actually make us think and remember. Now, in Alzheimer's disease, specific protein aggregates are formed in the brain, as you can see up here, so-called amyloid plaques, which over time is destructive for the nerve cells and cause them to lose contact with the neighboring nerve cells, and they start to shrink, shrivel, and eventually die off, which also cause the symptoms of the disease. Now, these specific key pathological hallmarks of the disease is what we target with our two projects, Alzstatin and NeuroRestore. Alzstatin targets the formation of these proteinaceous aggregates in the brain, these amyloid plaques, and NeuroRestore targets the neuronal dysfunction that results in the symptoms of the disease. Now, just to give you some figures of the disease itself, obviously, I think most of you have some type of relation with this disorder. It affects over 40 million people worldwide. And it's a 100% fatal disorder. Mean life expectancy after diagnosis is between 8 and 10 years. And the associated socioeconomic costs for this disorder are huge and approaching $1 trillion this year alone. And if we look at the available therapies in this area, we don't have much to offer the patients today. Only 4 drugs are available on the market today, 3 of which share the same mechanism of action and all share the same problems with limited efficacy, both with regard to size and duration of effect, as well as a number of side effects, limiting their use in the patients. So it's not unexpected that the market for a novel drug in this area is enormous. So how do we then go about working in this quite complex area? Well, for us, we're working on 4 different pillars to increase our probability to get all the way to the market. One is that we work with mechanisms that have a genetic linkage to disease, that validates the mechanism and increase the probability that we'll actually see an effect, a real effect in the patients. We're working with small molecule therapies, and we have seen in the area quite large amount of large molecule therapies, but certainly for brain disorders, we believe that there's a clear advantage of using small molecule therapies, which I'll get back to you later on. We're obviously also working a lot with biomarkers development, both for patient inclusion, but also with regard to efficacy measures. And as Homan mentioned, we had a key opinion leader event with Professor Henrik Zetterberg from the University of Gothenburg, but also UCL in London, who's a world authority in the area of biomarkers and diagnostics in this specific field. So it's a clear highlight of what we do in the research. Moreover, we are actually having more than just one candidate in our pipeline. So we're working with 5 different candidates in our product portfolio with different chemical characteristics, both as backup compounds, but also compounds that can be used for other indications that I'll come back to later on in the presentation. Now, this area has seen quite a lot of advancements, on the contrary of what some people may think. And just looking at the last 10 years, we have learned a lot with regard to our understanding on basic disease biology, but also with regard to diagnostics, biomarkers, and in this case, just showing genetic risk factors in the area. There's been quite a lot of an increased understanding in the basics of the disease. And this is obviously something that we bring along in the development of our new therapies. And we can bring along also with regard to the design of the clinical studies. As I mentioned, we're working specifically on small molecule therapies. Now, in this specific area, we've seen a lot of focus on immunotherapies, antibody-based therapies, and we've heard and obviously seen also the fantastic results coming out from the oncology area using antibodies. But certainly when it comes to treating disorders of the brain, they have a problem in that antibodies do not readily cross into the brain from the blood, so they do not readily cross the blood-brain barrier. Approximately 1 in 1,000 actually cross into the brain, which means that you need to have large amounts of antibodies pushed into the circulation to get sufficient quantities up to the brain, which in turn results in high cost, an increased risk of side effects, but also, obviously, being an invasive type of therapy, you need to take repeated intravenous injections. And here, a small molecule tablet is obviously a huge step forward, especially here since we're considering a lifelong treatment that in a huge population, then we need a cost-effective solution, and certainly a tablet is the preferred option. Just to highlight some advancements also in the biomarker field, and as you may know, previously we have been relying on spinal taps, so taking a spinal sample to look at the biomarker profile there to say whether a patient is at risk of developing Alzheimer's or if he's already developed Alzheimer's disease. Now, taking a spinal tap is both invasive, quite cumbersome, and quite expensive. And obviously a blood-based marker, as we've heard about today, the diagnostic fields and the focus on blood-based markers is certainly a huge step forward. Now, the last 2 years, both American and Japanese scientists have shown assays which show very good sensitivity and accuracy based on blood-based markers, which is a huge step forward, both when it comes to patient inclusion, but also with regard to using them for efficacy in the clinical trials. So this is a major involvement in the area, I would say, and actually been somewhat of a highlight at last year's Alzheimer's conference. Now, coming back to the team itself. So the people and the founders in AlzeCure are all from pharma industry. As you can see, shows up here, AstraZeneca comes back, I think, in all of us. So coming here is actually feeling like almost coming home, which is always fun. But again, we've all worked between 15 and 35 years in the industry, worked with both early, late-stage projects, in-licensing, strategic management, target generation, etc., and also taking products to the market. And we have complementing expertise, ranging and covering the value chain from early medicinal chemistry to regulatory submission. The board of directors consists of Thomas Poller as our chairman of the board, with an experience both in the life science industry, but also in the private equity side, working at 3i for many years. And we've also supplemented the board with a number of people with a clinical background. We have Ann-Fan S. Johansson, who's been working in clinical operations and global development many years, both at Roche and most lately at Sobi. Ragnar Linder, who's been working with marketing and strategic experience from the CNS dementia area at Höchst and Aventis, and most lately working actually at Quintiles. And Ellen Donnelly, which is a quite new addition to our board of directors, who until just a year or so ago worked at Pfizer, being responsible for their clinical portfolio in the neuroscience and pain area. And we also have Pirkko Tamsen, who's proposed to be elected as director of the board, and who has an experience working both at global clinical project lead in big pharma, as well as running clinical CROs, which is obviously our next step, and that's why her expertise is highly valued as well. As I mentioned earlier on, we're working with a number of key opinion leaders in the area, and this is obviously a key item for us because we need to get access to the latest developments in this specific area, both when it comes to disease biology, but certainly when it comes to design of clinical trials. So we're working with Professor Bengt Wiblad, who's a professor in geriatrics at Karolinska Institutet and one of the world's most published authors in the field just a few years ago. Professor Henrik Zetterberg, as Homan mentioned, we had a key opinion leader event with him just a week ago. And he's a world authority in the area of diagnostics and biomarkers in the field. And he made, I think, a very good presentation on the fantastic advancements made in that specific area. We're also working with John Harris and Pete Snyder, who both are experts in the design of the clinical studies, which are certainly key for us as we are now moving into these important clinical phases. Now then, let's move into the more fun part of it, obviously then the projects themselves. So as I mentioned, we have two different product platforms, the NeuroRestore and Alzstatin projects. And starting out with NeuroRestore, which is, as I mentioned, targeting the neuronal dysfunction in the disease. And in the normal brain, nerve cells communicate with each other in these vast networks, and actually interchange of information is here at this local synapse. And actually the loss of synapses early on in the disease is what correlates best with the symptoms. Now in the disease, there is a very poor signal communication between nerve cells, and NeuroRestore actually binds in the synapse to enhance neuronal communication, which we've been able to show. So how does NeuroRestore do this? Well, NeuroRestore targets a specific, highly interesting biological mechanism in the brain, actually received a Nobel Prize in the mid-80s, which is called the neurotrophin signaling system. And the most well-known are BDNF and NGF. Now these growth factors, they bind to specific receptors on the cell surface, which results in important functions in the cell, such as enhances the communication between nerve cells. It supports neuronal function, survival, and protection against external toxic insults. But even more importantly, also supports the function of the brain. It also supports cognitive function, and it plays a critical role actually in normal cognitive function. Now in Alzheimer's disease, there is a disruption of the system so that the signaling is suboptimal or subnormal, so it's lowered compared to normal situations. And what our compounds actually do is that they act here and enhance signaling through the system and restores normal signaling of the system, enhancing neuronal communication, which we've been able to show, as well as enhancing cognitive function, which we've also been able to show in multiple animal models and species. Interestingly, there is also a genetic linkage to the disease, as I mentioned earlier on. In fact, in about 30%, so certainly a number of us in here, have a mutation in this BDNF gene that results in a 30% decrease in the normal signaling of this system. Now, if you're healthy, it doesn't show that much. However, if you get Alzheimer's disease, you get a more rapid cognitive decline and a more aggressive pathology, indicating that this system is critically involved and important to be able for the neurons and the brain to survive the toxic insults of Alzheimer's disease. Moreover, this genetic linkage has also been seen in other indications besides Alzheimer's disease, also in Parkinson, traumatic brain injury, and sleep disorders, meaning that there are actually other indications that are also possible for this mechanism of action. Another interesting feature with this unique mechanism of action that we're targeting is that it has additive effects to existing therapies that are currently in the market. Now, this is a key selling point when coming to the marketplace because many of the current therapies will be generic at the time. And it's important for us to show that it has not any detrimental effects on those compounds, but in fact, we can show that it has additive effects to this. So it's a clear key advantage, I would say. Now, our lead compound, 855, which is the first in this class, is entering phase 1 clinical studies later this year. And as you can see, we also have a backup compound and another compound going for a different indication in the pipeline. Now, the interesting part here is that the phase 1 clinical studies, which are obviously aimed to look at safety in healthy volunteers, we have designed the phase 1 clinical program in an innovative way so that we will actually also be able to look at early efficacy signals already in the phase 1. Because we know from our past in the in-licensing side the importance of having early efficacy signals that can trigger interest from a third party. To the next project then, which is the alzestatin project, targeting then the formation of these amyloid plaques in the brain. Now, these amyloid plaques are formed by the sequential cleavage of a protein called APP. And this cleavage is caused by two enzymes or scissors. The first name is called beta secretase, and the other one is called gamma secretase. The resulting peptide is called amyloid beta, and this is a very sticky peptide. So it starts to aggregate if you get to sufficient concentrations and forming different forms, protofibrils, oligomers, and eventually these large-scale amyloid plaques. And these forms of amyloid are toxic for the nerve cells. So this is obviously something we want to prevent the formation of. Now, what alzestatin does is actually binding to this enzyme, the gamma secretase, and it causes the enzyme to, rather than cleave off this sticky A-beta peptide, actually forms smaller, short peptide fragments which are benign, do not aggregate, and actually enhances the clearance of these toxic peptides. So it actually has a dual action. Now, this project is actually an AstraZeneca legacy project, so we were part of initiating that project at the time at AstraZeneca, and also part of the project team. We were able to take this project forward and license it in, and have continued evolving these compounds further on. Now, this slide shows you what we just saw on the previous movie, actually the APP protein being cleaved by first beta secretase and then gamma secretase. Now, as you probably have seen in the area, there's been a lot of focus on inhibitors of this enzyme, the beta secretase, so-called BACE inhibitors. Now, we were part of developing AstraZeneca's BACE inhibitor that went into phase 2/3. However, we knew already at the time that there is a problem with this approach, because this enzyme not only cleaves this protein, but actually some 50 other proteins in the cell, some of which play a critical role for normal cell functioning. So the risk of side effects is large. It's therefore that we did develop a backup strategy at AstraZeneca at the time, developing compounds that instead targets this enzyme, but rather than using a sledgehammer approach, actually modulates the enzyme activity, so it only affects the cleavage of this specific peptide and does not touch the normal physiological function. Now, this was a huge undertaking at the time, and AstraZeneca spent some 200 million Swedish kronor into this project, even though it was a really early stage. We eventually found some really good compounds that does just this. Actually, instead of forming this sticky A-beta-42 peptide, forms these shorter peptide fragments which do not aggregate RB9 and actually helps to clear these toxic A-beta-42 peptides. Just to highlight very briefly as well, is this graph, which shows something that we didn't know some 15 years ago. We didn't know at that time that the disease actually starts 15 years before symptom onset. That also explains the reason for why we went into the disease very late without treatments and didn't see much of an effect. And just looking at the red curve, which is actually the amount of amyloid in the brain, you can imagine going in here that the chances of seeing effect is very, very small. So that's why the area is now moved up much earlier in the disease, as we've seen now with BioArctic's antibody. They came out this summer showing effects if you go in earlier in the disease. Very briefly to say that this compound has shown obviously very good effects on lowering these toxic peptides, both in cellular and animal models. I mention again, it's an inert mechanism that does not touch the normal physiological function of the enzyme, reducing risk for side effects. So it's certainly the treatment of choice if you're going for a lifelong treatment in the disease. It also has a strong genetic linkage to disease. Actually, 9 out of 10 with inherited forms of the disease have a mutation in this specific enzyme. And it has also clear differentiation compared to other products currently in development. And also here in this project, we'll actually be able to look at not only safety in the phase 1, but also early efficacy signals. We can actually measure biomarkers already in the healthy volunteers, so we'll be able to look at target engagement already in the phase 1. And again, here we have also backup compound. 679 is the first one. It's planned to enter clinical phase 1 in the end of next year, and followed by the backup compound 680. And again, this is my almost last slide, just showing you the BAN2401 data from BioArctic ASI Biogen that came out this summer that probably no one has missed, but obviously has played a very important role for patients, for the field, but also for us, because the BAN2401 data that came out showed that if you lower amyloid, and if you go early enough in the disease, then you will also have an effect on cognitive function. Now, this is important because it also validates the amyloid mechanism, which is also what we're targeting with alstatin. And my last slide is just some take-home messages that we are certainly targeting an area of huge unmet medical need with a huge market size. We do so with a strong team with extensive experience in this specific field. We are working with 2 different platforms targeting different patient segments, both symptomatic patients and earlier in the disease. We also have multiple candidates in the project pipeline, making us having more shots on goal and making it all the way to the market. Thank you very much for your attention.
Great.